if you get a deep cut on your arm or even just a bad scrape, there is this underlying expectation of a biological trade-off.
Right, yeah, the inevitable scar.
Yeah, exactly. We almost take it for granted. It is a bit like fixing a shattered ceramic mug. You can carefully glue all those pieces back together and it will hold your coffee again, but you are always going to see the cracks. The tissue knits back, but, it leaves a mark. It leaves a scar.
It's patched, yes, but it is certainly not the original fabric. For the longest time, we've just accepted that scarring, that rigid raised tissue, is simply the inevitable tax we pay for surviving a severe injury.
But then you step into the world of molecular dermatology and suddenly that whole trade-off concept is just thrown completely out the window. We are looking at a biological landscape today for this deep dive that is pulling off what seems like, well, an impossible magic trick.
Regenerating skin without the scar.
Yes, true regeneration. And we are pulling from a massive stack of research today to figure out how this actually works. We're looking at marine biology, specifically polynucleotides sourced from salmon DNA.
Right, the salmon gonads, which is fascinating.
Yeah, and we're crossing that with botany, looking at plant-derived polynucleotides from things like the Peonia lactiflora and the snow lotus, and of course diving deep into human cellular signaling. But for you listening, this isn't just abstract science. This is the literal blueprint of how your own skin heals, how it ages, and how it might actually be able to rebuild itself perfectly.
And the central mystery we are unspooling here is quite specific. How do these DNA fragments, these polynucleotides, command your skin to rapidly produce fresh, youthful collagen, specifically type I and type 3, without, triggering that chaotic fibrotic mess we know as a scar?
Okay, let's unpack this, because if I introduce foreign DNA, like from a plant or a fish, into my skin, my first instinct is that my body should treat it like an invader, right? Why does this regenerative process actually work instead of causing a massive immune reaction?
That is exactly the right question to start with, because it comes down to a process called cellular recycling. Polynucleotides, or PNs, are essentially just highly purified fragments of DNA.
Okay.
So it doesn't actually matter if they're sourced from salmon or specific plant calyces. At this purified fragment level, they are universally biocompatible. When introduced into human tissue, the body doesn't see an invader, it sees raw materials.
So it's like dropping off a truckload of bricks at a construction site.
Precisely. The cells utilize something known as the salvage pathway. Normally, if a cell wants to repair itself or multiply, it has to build new DNA from scratch, molecule by molecule.
Which I assume takes a lot of energy.
A tremendous amount of biological energy. But with the salvage pathway, the cell just scavenges these pre-made polynucleotide fragments that we've introduced.
Wow, okay, so it's a massive energy-saving shortcut for the skin cells. But from reading the research, it seems like their structural role, just being spare parts, isn't even the main event.
It's really not. The true magic happens when these PNs interact with the surface of human dermal fibroblasts. Fibroblasts are the factories in your skin that pump out collagen.
Right.
And on the surface of these factories, there are specific receptors waiting for instructions. One of the most important ones is called the adenosine A2A receptor, or A2AR.
And the sources heavily emphasize this receptor. It seems like it's the master thermostat for tissue repair, but how does it actually communicate with the inside of the cell?
What's fascinating here is how delicate this communication network really is. When the polynucleotides bind to this A2A receptor, they trigger the release of a messenger molecule inside the cell called cyclic AMP, or CAM-MP.
Okay.
Think of CAM-MP as the internal factory manager, taking the order from the receptor and telling the workers what to do.
Okay, and this leads to what the researchers call the CAM-MP paradox.
Uh-huh.
I got a little lost in the data here because it seems like giving the factory manager an instruction can either lead to perfect healing or a total shutdown, depending entirely on the dosage of the signal, right?
That's exactly it. When PNs bind to the receptor, they create a very modest, controlled increase in that CAM-MP messenger. We are talking about a gentle nudge, roughly a 150% increase above the cell's normal baseline.
Which sounds like a big jump, 150%, but in cellular biology, that's just a whisper, right?
It is a whisper. It's a physiological nudge that says, hey, it's time to wake up, start multiplying, and start synthesizing collagen via these non-canonical AKT-dependent pathways.
Right.
But, and this is the crux of the paradox, if you bypass that delicate receptor and force the cell to produce too much CAMP, The entire system crashes.
The sources mention scientists running a stress test to prove this. They used a chemical, I think it was called forskolin.
Yes, for Skolin. They used it as a direct activator to artificially force the cell to produce massive amounts of CAMP, totally bypassing the natural checks and balances.
Yeah.
We're talking about a 15,000% spike in that messenger molecule.
Oh, wow. 15,000.
Yes, and the result, extreme biological panic, actually inhibits type I collagen production and completely stops the cells from proliferating.
It's like the A2A receptor is a highly sensitive volume dial on a high-end stereo, rather than just a simple on-off switch. If we crank the volume to the absolute maximum to that 15,000% spike, the speakers just blow out. The music stops. The factory shuts down.
Right.
But at the perfect mid-volume, that gentle 150% nudge, the factory hums along beautifully. Is that why polynucleotides don't cause the hyperproliferation we see in fibrosis?
What's fascinating here is that precise dose-dependent signaling is the exact first vital step in avoiding scar tissue. Fibrosis, which is what a scar is often the result of cellular panic. It's a biological system running at maximum capacity with the brakes cut.
Wow.
By keeping the signaling strictly within that physiological sweet spot, polynucleotides initiate a robust healing response without ever tripping the alarm bells that lead to chaotic, overgrown scar tissue.
Okay, so the factory is running at the perfect speed. The workers aren't panicking. The volume is dialed in. Let's look at the actual output. What exactly is this factory producing? Because the shift in the types of collagen being built is really fascinating.
To understand what is happening, we have to look at the mechanics of the two primary structural proteins in your skin, type I and type 3 collagen.
Right. And usually, if you just look at healthy adult skin, it sits at a pretty stable ratio. You have about 80% type I collagen and 20% type 3. But when these specific short-chain polynucleotides are introduced, that ratio completely shifts, doesn't it?
It does.
The fibroblasts go into overdrive and start aggressively pumping out type 3 collagen. The researchers affectionately call it baby collagen. And within just 48 hours, it physically shifts the environment in the skin from 80-20 down to 60-40.
And that type 3 baby collagen is very distinct. It's extremely thin, only about 30 nanometers thick, and highly flexible. It is designed to be deployed incredibly fast. But to build it at that speed, the internal environment of the cell has to be absolutely flawless.
Yeah, the research got very specific here about the manufacturing floor inside the cell, right? The endoplasmic reticulum.
Yes, inside the endoplasmic reticulum, the environment has to act like a perfectly balanced clean room. The pH has to be perfectly dialed in at 7.1 to 7.2.
Oh, that is specific.
Very. And the cell begins heavily recruiting raw materials, specifically localized vitamin C, roughly 50 micromolar of it, to act as a crucial catalyst.
Wait, I want to pause on the vitamin C. I mean, we all put it in our skin serums, but what is it actually doing mechanically inside the cell at that exact moment?
It acts as the physical mechanism that allows the collagen to take its shape. Collagen starts as 3 separate loose polypeptide chains. The vitamin C catalyzes a reaction that forces those 3 threads to coil themselves tightly together into a triple helix.
Oh, interesting.
Yeah, without it, the coil falls apart. That tightly wound, successful helical coil is what we call procollagen.
And the researchers actually watched this happen in real time, didn't they? They tagged the molecules with carbon-14 isotopes so they could track exactly where the raw materials were going.
They did. And what the carbon-14 tracking revealed was astonishing. Once the PNs give the signal, the cells begin hoarding the raw amino acids required to build this collagen at an unprecedented rate. The concentration of hydroxyproline, a key marker, jumps from 15 micrograms per milliliter to 21.3 micrograms per milliliter by day 14.
They're essentially stockpiling bricks to build the wall as fast as possible. And the sources draw a really sharp contrast here between how the short fragments of DNA act versus the longer fragments, right?
Yeah, right.
The short chains are speed running the process, consuming 45 micrograms of free amino acids every single hour just to throw up that flexible type 3 baby collagen. But the long chains, they take a totally different, much slower approach.
Yes, the long chains retain their length to slowly replace type I collagen at a rate of just 2% a day, laying down much heavier 65 nanometer thick bundles over the course of weeks. If we connect this to the bigger picture, it is all about structural timing.
It reminds me of disaster recovery construction. Let's say, right after a major earthquake. The short DNA chains are acting like the emergency crews. They throw up rapid, flexible scaffolding, the type 3 collagen, immediately to stabilize the structure.
Right.
It's fast, it's pliable, it holds the building up so it doesn't collapse, but you can't leave temporary scaffolding up forever. So the long chains act like the heavy-duty construction crews. They bring in the permanent steel beams, the type I collagen, slowly over 28 days to provide lasting structural tension.
And that tension is mathematically crucial. We're talking about achieving a lasting structural tension of 2.5 millinewtons per meter. That mechanical tension is literally what gives youthful, healthy skin its snap, its ability to bounce back.
But here's where it gets really interesting. I mean, we are talking about a massive influx of material. If the skin is stockpiling amino acids, pumping out huge amounts of scaffolding, and bringing in heavy steel beams at breakneck speeds, why doesn't it just result in a biological traffic jam?
Right.
Why doesn't the skin overproduce and form a raised keloid scar?
That is the multi-million-dollar question in regenerative medicine. If we are building so much, why isn't there a scar? And the answer lies entirely in how these polynucleotides tame the inflammatory fire.
Explain that, because we usually think of inflammation as a normal part of healing. You get a cut, or, it gets red and swollen.
It is normal, but it's also the primary driver of fibrosis.
Huh.
A scar isn't just a result of making too much collagen. It's driven by chronic, unchecked inflammation that leads to the chaotic degradation of the extracellular matrix. It's the body panicking.
Okay.
Polynucleotides actively suppress those initial panic signals. They inhibit mast cell degranulation and reduce the nuclear localization of NF-kappa B.
So they stop the panic at the source.
Exactly. By doing that, they prevent the release of destructive inflammatory cytokines like TNF-alpha. The body knows it needs to repair, but the inflammatory fire is starved of oxygen.
Okay, I followed that. But there is a fascinating scientific debate in the research that I want to bring up. Because as I was reading the notes on the different sources, the plant-derived PNs versus synthetic ones, I got a little lost. It seems like the research is slightly split on how this scar-free guarantee is actually achieved.
Yes, this is a beautiful example of how nuanced molecular biology can be depending on the source material. Some studies looked at a synthetic version of the receptor signal, CGS 21680.
Okay, the synthetic one.
Right. They found that collagen production happened completely independent of a very specific pathway known as the SMAD23 pathway.
And just to clarify, from what I read, turning on the SMAD pathway usually means you get a scar, right? It's deeply linked to fibrotic tissue.
Exactly. SMAD signaling is a notorious driver of both cardiac and dermal fibrosis, so if the synthetic signal avoids it entirely, it makes logical sense why there's no scar. But here's the twist. The research on the plant-derived polynucleotides, PLPN, specifically from the Peonia lactiflora flower, showed something totally different. The plant DNA actually does activate TGF-beta and that notorious SMAD23 pathway.
Right, and that's what confused me. If the plant DNA is turning on the pathway famous for causing massive scars, how are the clinical results showing beautiful, smooth skin with absolutely no scarring? Like, how can a profibrotic pathway be activated without leaving a scar?
This raises an important point about the cellular environment. It's not just about which switches are flipped, it's about the counterbalances in the room. And the answer to your question comes down to a very specific biological tug-of-war between two elements, the TIMP1 to MMP1 ratio.
Okay, you're going to have to translate those acronyms for me.
We'll start with MMPs, matrix metalloproteinases. Think of them as biological scissors.
Biological scissors, got it.
During normal, messy healing, these scissors haphazardly chew up the damaged, torn tissue to clear space for the new material. But in fibrosis, when the body is panicking, those scissors go rogue. They start snipping everything, causing chaotic breakdown and a very messy, tangled repair job.
Okay, rogue scissors causing a mess. What is the counterbalance?
TIMP1. It is the tissue inhibitor of those scissors. Think of it as the protective safety guard that slides over the blades.
So what do the polynucleotides do to this tug-of-war?
Normally, during an injury, the ratio of safety guards to scissors is about 1-to-1. It's a chaotic free-for-all. As fast as you build, things are getting chewed up. But the PNs actively downregulate those destructive scissors, specifically MMPs 1, 3, 9, and 13, while significantly elevating the safety guards.
Oh, wow.
They change the protective TIMP1 to MMP1 ratio from 1-to-1 up to 2.5-to-1.
Wow, so there are 2.5 times more safety guards in the air than there are scissors.
Exactly. So even if that notorious SMAD pathway is driving incredibly fast, robust collagen production, the elevated safety guards ensure the environment remains highly controlled. It enforces a pro-repair state that strongly favors laying down a durable extracellular matrix rather than getting caught in that messy fibrotic cycle of chaotic breakdown and rushed repair.
That is incredibly elegant. The body is building fast, but the environment is so heavily guarded that no mistakes are allowed. But, making the collagen and preventing the chaotic breakdown is still only half the battle, isn't it? Because if you have all these new type by steel beams and type 3 scaffolding fibers just floating around in the skin, how does it finalize the process? How does the tissue organize them so the surface of your skin actually looks smooth and feels elastic, rather than feeling rigid and bumpy?
This brings us to the remodeling phase. And it's important to note this doesn't just happen overnight. The scaffolding goes up fast, but the remodeling extends from about day 21 deep into the months that follow. And the hero of this finishing phase is an enzyme called lysyl oxidase.
And what does this enzyme actually do?
As its concentration climbs to a peak of 45 nanograms per milliliter, lysyl oxidase utilizes copper ions to stabilize the newly formed, scattered collagen networks. Think of lysyl oxidase like a skilled old welder operating just beneath your epidermis.
Okay, I like the welder visual. So what are they welding?
You have all these loose connection points or nodes on the newly laid collagen fibers. This master welder comes in and systematically ties off those loose ends, forging them into highly stable, organized crosslinks. And crucially, because we suppressed all that inflammatory panic earlier, this welder is operating in a very calm environment. It aligns all the microscopic collagen fibers perfectly along the natural mechanical stress lines of your skin,
As opposed to what?
as opposed to a scar, where the collagen fibers are just a tangled, randomized web of rigid tissue piled on top of each other.
So what does this all mean for you, the listener? When the welder is done, when the safety guards have done their job, what is the concrete physical result? Well, the researchers actually measured it. Using incredibly precise instruments to gauge the tensile resistance of human skin, they found a 14% absolute increase in surface elasticity.
Yes, the R2 value, that
Yeah.
is a highly significant structural improvement.
Right. A 14% increase in the physical bounce and resilience of the skin. And that isn't from pumping the skin full of artificial fillers. It is generated entirely by this highly organized, naturally reprogrammed, scar-free remodeling process.
It really is a masterpiece of biological orchestration.
It's just mind-blowing. Okay, let's take a step back and recap this incredible journey we've been on today. Because it's a lot of biology, but the sequence is beautiful. It starts with those polynucleotides, whether from the ocean or a plant, hitting the A2A receptor thermostat.
Right.
That receptor acts as a highly sensitive volume dial, giving the cellular factory a gentle 150% physiological nudge, not a panic-inducing spike.
Which initiates the rapid deployment of the flexible type 3 scaffolding, eventually replaced by the strong type I structural beams.
Right. And while all that building is happening, the DNA fragments are actively suppressing the inflammatory fires. They keep the panic messengers out of the nucleus, and they flood the area with safety guards to stop the biological scissors from chewing up the new tissue. And finally, the master welder, lysyl oxidase, comes in, locking those permanent steel beams into a perfectly smooth, elastic network aligned with the natural pull of the skin.
And if we connect this back to you, the listener, it's worth remembering that this intricate, invisible ballet is capable of happening continuously, just millimeters beneath your skin. It proves that true regeneration isn't just about raw production, just pumping out collagen. It's about precision, timing, and an exquisite balance of molecular forces.
It completely changes how you look at the concept of healing. You don't have to just patch the ceramic mug anymore. But looking at all of this leaves me with one final kind of mind-expanding thought. We've been intensely focused on dermatology and skin today, but if we can bioengineer these specific chain links of polynucleotides to perfectly orchestrate this scarless tissue repair on the outside of our bodies, well, could we eventually deploy targeted PDRN therapies to reverse life-threatening fibrotic scarring deep inside our bodies?
Oh, absolutely. The underlying mechanisms of fibrosis, the inflammation, the cellular panic, the chaotic collagen, they are remarkably similar across different organs. If we can master this receptor signaling in the dermis, the implications for internal medicine are profound.
I mean, imagine repairing the rigid, scarred tissue of a heart after a massive heart attack, or regenerating fibrotic lungs simply by giving the cells the right molecular nudge to rebuild perfectly instead of panicking. It's an incredible future to mull over. Thank you for joining us on this deep dive. Keep questioning the amazing mechanics of the world around you, and until next time, keep exploring.